Microfluidic devices, they are basically microchips that contain channels that you could culture cells in. So you would basically add normal media, for example, differentiation media, dedifferentiation media, and basically you would culture it as in a Petri dish, and it would work perfectly. So, so we start with targeted differentiation. So for differentiation of stem cells, you start from a totipotent fertilized zygote, and then there's the normal process of embryonic development. And then you use various transcription factors which activate genes, and that would lead to gradual speciation and production of many cell types. And for example, these would be the transcription factors. And obviously, this might give rise to a chicken and egg problem, because for the transcription factor to exist, it needs to be expressed by the gene in the first place. But there are maternal transcription factors that exist in the zygote, and this is important later when we consider the experiments that led to dedifferentiation being recognized. And so you can basically think of the process of differentiation as being like a tree and its branches. So you branch and branch from its stem, and there is a hierarchy of transcription factors. So you would have the master transcription factors at the top, and it is obviously very dangerous for the activation of all the transcription factors at once, and the absence of certain transcription factors leads the cells to pursue an alternative pathway. So this would also reduce the need for a transcription factor for, say, every gene to be expressed. So we start off with the master transcription factors, which will be said on the next slide, and super enhancer elements, which carry clusters of enhancers and that are densely occupied by master regulators and mediator. But the details I will not go into here. So dedifferentiation is the opposite process of our differentiation, and it is important because even if you produce a microfluidic device, that would only be a model, but that would only be using cells from a cell line. So for it to have any therapeutic application, you would need to dedifferentiate from human epithelial cells and then produce induce pluripotent stem cells before you differentiate again to form cells of various types. And that would be much less invasive than, say, a biopsy. And so I've circled that compound reversine up here, and that is a compound that causes the reversal from a unipotent state to a more to a less differentiated state, but that is not enough to get us back to our pluripotent state. And to do that, we refer to the experiments of somatic cell nuclear transfer and basically apoptotic genes, which were done by Gurdon and Yamanaka a very long time ago, which led to them being awarded the 2012 Nobel Prize. So Yamanaka basically deduced that many of the relevant reprogramming factors would be expressed predominantly in the eggs and embryonic stem cells. And so he compared the transcriptome and proteome of these stem cells and somatic tissues. And he found that there were indeed embryonic stem cell specific markers. And then he generated knockout mice to examine the phenotypic effects of such genes, and they identified 24 reprogramming factors, but even these homozygous mutant cell lines did not yield any drastic phenotypes or lead to early lethality. And the clever step was to generate a strain whose somatic subtype was susceptible to a compound, but their stem cell population would be resistant to high concentrations of this compound, which meant that successful reprogramming would allow the cells that had undergone reprogramming to survive and make ES cell colonies. So none of the 24 candidates allowed the obtaining of drug-resistant colonies alone, but combinations thereof eventually did. And this combination right here, that's the simplest and smallest number of transcription factors needed, and they are known as the Yamanaka factors. The mechanisms governing this process are relatively unknown, but I would suspect that there are complex feedback processes involved in this, and there are transcription factors somehow turn off more lineage-specific transcription factors through novel mechanisms. So we now go to recent papers that have been published in this subject. And the two on the left come from the lab of Professor Donald E. Ingber at Harvard in the Department of Genetics. So the first one involves the reconstitution of kidney glomerular capillary wall function on a chip, and they were eventually able to demonstrate differential clearance of insulin and albumin, which is a very important factor that demonstrates recapitulation of liver function. More importantly, it was able to mimic adriamycin-induced albuminuria and podocyte injury with fluid flow and cyclic mechanical strains showing that these were essential for the formation of organ-level structures that could closely resemble the normal tissue-tissue interface. And the second paper, which I have taken some extracts from here, showed that basically, they show that breathing is heavily associated with how far the tumor could spread. And the growing cancer cells, which are accompanied with a loss of lung motion due to filling of the alveolar spaces, would have a very large positive feedback loop and further enhance tumor cell proliferation during disease progression. And the diagram on the right is very interesting because it is one step towards the building of a human on a chip. So the group was trying to study the absorption, distribution, metabolism, elimination, and toxicity, basically pharmacokinetic factors in a microfluidic device rather than in a animal pharmacology testing. And basically, it shows that when a toxicant was introduced into the microchip, organs, including the liver, the lungs, they all showed phenotypic effects that would have been expected from these. And these go very well in showing that a human on a chip is in fact possible, but as of now, we are working well onto this. And so the applications of this technology are profound. These two have already been mentioned. And the vaccine development, I am not entirely sure about, but I will be meeting with Professor Ingber next Monday when he comes to the Royal Society for a keynote lecture. Hopefully that will be resolved. I will update you if anything exciting comes from that. And the microchips are translucent, and obviously they would be able to show how human organs can work, but at this current stage, we're not able to model properly enough to see if we could make further improvements based on this. And there have been entrepreneurial setups from this company, reduction in cost, reduction in animal testing is always a good thing. And my research project in the summer involves the epithelial to mesenchymal transition using hepatocytes. And the drawback to this is that there are very limited differentiation protocol for these cell types. They reach about 80% and they stop there. And we're looking for about 90% to actually do anything productive. And if successful, we're hoping to use this as a drug platform. So we could potentially try to analyze if folk medicines are like therapeutically they have efficacy. And so because many of these are well rooted in basic science, for example, willow tree bark and containing aspirin and penicillin or ginseng, we could use these and see if the ETM is reduced. And with that, I conclude my talk. Thank you.